In the last lecture, we have discussed how the blood group is being decided. A, B, AB or O. If you have not gone through the first video, please go through the first video. Then this second video of connected to the blood group will help you. Now, if a blood group is containing antigens, then it is also containing antibodies. It is also containing antibodies. What are antibodies? Now, antigens are glycoproteins. Glycoprotein. Protein means a peptide chain, and glyco means it is also having polysaccharides. Now, antibodies are basically proteins. So, blood group is decided by antigens, but it also contains antibodies. Which blood group contains which kind of antibodies has to be decided. To understand it, we have to understand key and lock system. Like in a particular lock, there is a specific key which is fixed. No other key can be fixed into any lock. So in that case, we have the same kind of process which is occurring inside the blood. This is called key lock system. Let us understand. Now we know that we have antigen on the surface of the RBC. We have what we have? We have antigen. This is hemoglobin which is present in it. We know that. We already have discussed about the hemoglobin, how much hemoglobin in single RBC is present. This is also containing what? Antigen. The antigen which is resembling with the A-like structure is called as antigen A, and it makes a blood group A. Now, how to decide which kind of antibody is present in this particular blood group which is containing antigen A? To understand it, let us make a little big diagram to understand key lock system. If antigen A is present, so it is having a triangular shape, correct? Present on the surface of RBCs. I am just making a bigger structure of antigen. Now, if it is having any antibody in it, let's make the diagram of antibody. Antibody is basically having this kind of structure. This is not elaborated structure, but to understand blood group, it will help us. If this antibody is there, it is also having an attachment point. If it is having this kind of attachment point, which also resemble the triangle of the antigen, then the antigen will get fixed into this particular A-shaped structure or A-shaped attachment point of the antibody. Again, remember, this is antibody and this is antigen. So, if antigen A is present on the surface of RBCs and antibody is also A, then what will happen? This A antibody is having triangular shaped depression. Then this antigen will fix into the A-shaped depression of the antibody, and what will happen? This antibody will get attached. Antigen A, this is antigen A which is having triangular shaped structure, and antibody is also having A-like depression. Then this antigen of RBC will get fixed into the antibody which is having A-shaped structure or antibody A. Then what will happen? This RBC is flowing into the blood. If this protein is get attached to this antigen, the flow of the RBCs will be restricted. If 2, 3, 4 antibodies get attached to antigens of RBCs, then RBCs will not float properly. Ultimately, they come on the junction of capillaries and actually a clot will form, that is called coagulation of the blood. So one thing is very much clear: if there is antigen A, then antibody A should not be present. A should not be present in the blood group. So which kind of antibody has to be present? Suppose I make a little change in it. I make a little change. Instead of this A-shaped structure, if it is having, antibody is having a B-shape, dome of the B, then antigen A will not get fixed in it. Antibody will, if come in contact, it will not get fixed, rather antibody will float over it, and RBCs, which is having this particular antibody, will also float very easily. Correct? So blood having RBCs having antigen A should not have antibody B, because key and lock system will prevail, and if antigen A is there, then the antibody B should be there, because B is having attachment point like a B or dome shape, which will not fix the triangle of A. So both will float very easily into the blood. So that is why if there is antigen A on RBCs of a blood, which is making blood group A, always will have what? Will always have antibody B in it. Antibodies are written with the small letter to avoid the confusion. So blood group A always contains antibody B, so that key and lock system should not prevail. And on the same way, if there is antigen B which is present on RBCs, which makes the blood group B, always should have what? Always should have antibody A. So key and lock system means this A triangular shape antibody will not fix into the dome of the antibody of B on the attachment point. So both will flow in the plasma. The same way, if this blood group AB simply means this blood group is containing antigen A and antigen B. This is having no antibodies, because there are two types of antibodies, A and B, and it is having A antigen as well as B. कोई भी अगर एक प्रेजेंट होता वो अटैचमेंट हो चुका होता, इसीलिए नो एंटीबॉडीज आर प्रेजेंट in blood group what? Blood group AB. On the other hand, if we have blood group O is having no antigens. Already in the last lecture we discussed, if there is no antigens present, so it is having both the antibodies. It contains antibodies A as well as B. Correct? So this way we can understand perfectly which antigen is present in the blood group and which antibody should be present logically in that blood group, so that coagulation should not take place. This is how antigen and antibodies are being decided in the blood group. I hope it helps you. Thanks.